Hi students, welcome to the Chapter 14 lecture. This lecture is going to be covering the brain as well as the cranial nerves. So we're going to be studying both the anatomy and physiology of the brain itself. And this is a fairly lengthy lecture. There are a lot of images and diagrams, however. So it's a lot of slides, but a lot of it is pretty picture heavy. So I'm going to go ahead and get started with a brief introduction. The human brain, of course, is extremely complex. That should go without saying. Brain function literally is associated clinically with what it means to be alive or dead. And the importance of the brain hasn't always been well understood. So Aristotle thought that the brain just cooled blood, whereas Hippocrates had a more accurate view of the brain's importance and functions. So what this chapter is setting out to do really is provide you with an overview of the anatomy and basic physiology of the brain and cranial nerves, which are directly connected to it. So some of the functions that we'll be considering with this chapter are motor control, sensation, emotion, and thought. Let's look briefly at how the brain develops embryonically. The nervous system, or the NS as it's abbreviated here, develops from a tissue type known as the ectoderm. So ecto basically means outer, derm is referring to skin. So this is the outermost tissue layer of an embryo. And there are some fairly good examples of this process illustrated and visualized in various videos on the internet. So I'll be linking to several of those such that you can take a look at those as well. But there's a structure known as the neural tube, which is apparent by day 26, and that eventually forms 3 anterior dilations that are going to correspond to the hindbrain, the midbrain, and the forebrain. Now, a dilation is basically just a swelling. So those are going to form the major parts of the brain, this basic neural tube that is to develop into our brain. So these 3 are going to further differentiate into 5 secondary vesicles by week 5. So we have the forebrain developing into the telencephalon. The lateral outgrowths of the structure become the 2 cerebral hemispheres, so the right and left portions of the main part of the brain. And then the diencephalon, which are cup-like optic vesicles that become the retinas of the eyes. The midbrain is known as the mesencephalon, and then the hindbrain consists of the metencephalon as well as the myelencephalon. So if we take a look at this image here, we have an embryo at 4 weeks of age, and all of these are consistently color-coded here. So we basically have this very basic structure that comes out of the neural tube dilations here. You can see there's a fairly simple linear design to these different tissue types. 5 weeks, we have a little bit more development of the myelencephalon, which is this part here. And then a fully developed brain is going to be a very distinct, highly organized structure. So we have the myelencephalon that has become the medulla oblongata, part of the brainstem here. Inferior to that is the spinal cord. Then we have the metencephalon, which makes up the hindbrain, consisting of the more superior aspects of the brainstem, the pons, for example. The cerebellum, which is often referred to as the little brain, is seen here. And then the forebrain is going to consist of the diencephalon and the telencephalon. So the forebrain is what you typically think of a human brain, you think of the forebrain. This is known as the cerebrum, so it is the major, largest portion of the brain, and of course it is divided into lobes that correspond with bones of the skull, such as the frontal lobe, which has most of our more complex functions residing in that region of the brain. Let's look at some major landmarks of the brain. And when we do our brain dissection, these landmarks should be fairly apparent. So in class, we do a brain dissection of a sheep brain, And of course, there are going to be obvious differences between a sheep brain and a human brain, but the directional terms are still going to be relevant. The major parts of the brain, such as the cerebellum, cerebrum, brainstem, all of that is going to be the same. It's just going to be a size difference. And there's a little bit of structural difference in terms of the way the brain is laid out, which makes sense for reasons that I will talk about during that dissection. Okay, so there's a couple of directional terms that you might want to be aware of here. The rostral directional term just means toward the forehead. Technically, it means towards the nose. So the rostrum is the nose, but towards the forehead or towards the front of the anterior of the body, for example. And then caudal means toward the spinal cord. And in quadrupeds, animals that use forelegs to move around on, that technically means toward the tail. The caudal fin, for example, in fish is toward the tail. So caudal, in the case of humans, is going to be toward the spinal cord because we are bipedal and we don't really have as much of an elongation to the brain that we see in other quadrupeds. The weight of a mature human brain is approximately 3.5 pounds in men, a little bit less in women. And that doesn't mean that men are inherently more intelligent than women. That just means that there's a correlation between body size and brain size, which of course makes sense. So we see that pattern in a lot of different areas of human anatomy. For example, with the average volume of blood is going to be larger or greater in men than women. Again, that just corresponds with average body size. So our human brain here, this is going to be rostral, so this would be basically at the forehead here, caudal towards the back of the head, and you can see this part of the brain known as the cerebellum is inferior and caudal to the main part of the cerebrum, and then we have the brainstem basically being all the components of the brain minus the cerebrum and cerebellum. Okay, so the cerebrum is about 83% of brain volume, so definitely the major portion of the brain. It consists of two cerebral hemispheres, left and right hemispheres, which are separated by a fissure or a gap known as the longitudinal fissure. The folds are known as gyri. So the kind of bulbous folded portions of the brain are the gyri, and then the recessed areas between those folds are the sulci. And then the corpus callosum is a structure comprised mainly of axons of neurons that connect the left and right hemispheres of the brain. Then we have the cerebellum. Cerebellum is considerably smaller than the cerebrum, but somewhat surprisingly, it contains about half of the brain's neurons. It's of course the second largest brain region, and it's located in the posterior cranial fossa, which is a landmark that we learned when we learned about the skeletal system. So if you have a base of a cranium or a cranial model in your hand, you can easily see this posterior cranial fossa and how the cerebellum fits into that space. And then finally, the brainstem is simply the portion of the brain that remains if you were to remove the cerebrum and the cerebellum. So the diencephalon, the midbrain, the pons, and the medulla oblongata, all of which we will be discussing. So another view of the three major portions of the brain. These images were taken from Anatomy and Physiology Revealed or Cadaver Dissection Tool. So we have the brainstem here, the cerebrum, and then the cerebellum. And we'll take a look at the corpus callosum separately, but this large, kind of irregularly shaped structure here is the corpus callosum. Okay, let's look at a superior view of the cerebrum. So the longitudinal fissure, as I mentioned, is basically the gap between the left and right hemispheres. So it basically divides the cerebrum into two equal halves. It is a very deep gap. So we'll actually be doing a mid-sagittal section of our sheep brains, which means we're going to be separating the left and right hemispheres. We'll just be cutting down that longitudinal fissure, and you'll see that you'll get to the corpus callosum by making that cut, and you'll have two equal halves. The gyri, as I mentioned, are these thick folds, and then the sulci are these recessed areas between the folds, the grooves between the folds. Now, think about the functionality of having this highly folded structure. I want you to be thinking about some of the advantages of having a highly folded structure to the cerebrum. What functionality does that impart for nervous system functions? Compare that to hypothetically just having a smooth surface, having a brain that doesn't have these folds and grooves. And then the corpus callosum, as I mentioned, it's a thick nerve bundle at the bottom of the longitudinal fissure that connects those two hemispheres, so it provides for communication between the left and right hemisphere. So the longitudinal fissure highlighted in that orange color there, and then the gyri and the sulci. And corpus callosum, various views of the corpus callosum here. We have a lateral view. So this structure highlighted in orange here. Here is a coronal view or a frontal view. So you can see here that the corpus callosum does indeed actually connect the left and right hemispheres. Some medical imaging, as well as a transverse view showing again the connectivity between left and right hemispheres. So the major landmarks of the cerebellum, as I mentioned previously, the cerebellum does occupy that posterior cranial fossa within the skull. It also has gyri, sulci, and fissures. The structures that make up those folds just look a little bit different. So it almost looks like little leaflets. So the gyri are really kind of thin, almost like pages in a book that are stacked one on top of the other. The space between the cerebrum and the cerebellum is known as the transverse cerebral fissure. It is about 10% of brain volume, and as I mentioned previously, it contains over 50% of brain neurons. And then finally, the brainstem. Again, what remains of the brain if the cerebrum and the cerebellum are removed? The major components being the diencephalon, the midbrain, the pons, and the medulla oblongata, which is oftentimes just referred to as the medulla. Okay, so for components of the brainstem here, we have our diencephalon, our midbrain, our pons, and our medulla oblongata. Medulla and pons are fairly easy to recognize. Basically, the point at which the spinal cord enters the cranium at the foramen magnum. As soon as the spinal cord enters the cranium, that structure then becomes the medulla oblongata. So it's this first kind of subtly enlarged area of the brainstem here. The pons is going to be superior to the medulla, and it is the more pronounced swelling. The midbrain is going to be a very small area that's superior to the pons. And then the diencephalon is going to be this kind of crescent-shaped structure, superior to the midbrain, but inferior to the corpus callosum. So oftentimes with these brain regions, there's not necessarily a clear differentiation between, say, the midbrain and the diencephalon. You're just relying on relative location and landmarks to be able to identify those in, say, the sheep brain dissection. So looking at a lateral view of an illustrated brain here, this would be if we were to make a mid-sagittal cut. So cut right down that longitudinal fissure. We would see our corpus callosum That's made up of what's known as white matter. It's a white matter because it's highly myelinated, so it's mostly axons. You'll see the various lobes of the brain illustrated here, so frontal lobe, parietal lobe, occipital lobe, etc. And those largely correspond to the bones of the cranium that we learned. And we'll be learning about all of these structures that are listed here, but for now, just get a basic orientation down of relative size of cerebrum versus cerebellum, as well as how the brainstem fits into this picture. And then an actual image of a human brain here. It really illustrates the sulci, or the sulci, and the gyri very nicely. So notice just how deep these folds are. And again, just be thinking about what the advantage of having that highly folded tissue would be in terms of functionality. Okay, let's look at gray and white matter. You've probably heard these terms applied to brain tissue before and maybe not realize what it is implying. Gray matter is going to be where your neurosomas, your dendrites, and the majority of your synapses are. So neurosomas, remember, those are the cell bodies of neurons. Cell bodies of neurons. The dendrites are the tree branch-like extensions that come off of the neurosoma, and then the synapses are all of the areas of your neurons that are connecting with other neurons. So that's where your gray matter is going to be. It has a dull color that is kind of a grayish brown due to its lack of myelin. Now remember that myelin forms an interrupted sheath around the axon of both central nervous system neurons and peripheral nervous system neurons. The gray matter forms a surface layer, which is known as the cerebral cortex, over the cerebrum, and then a cortex also over the cerebellum. So the cerebral cortex is something that will come up a lot with this lecture because that's where a lot of our higher functions are. That's like the most complex functions to the human nervous system lie within that very thin outer layer on the cerebrum. Gray matter also forms nuclei deep within the brain, and nuclei would be plural of nucleus, of course. We're not talking about a single cell nucleus or an atomic nucleus, though. We're talking about organized centers within the brain that oversee a common function. So I'll be talking more about that later in this lecture as well. And then we have our white matter. These are comprised of bundles of axons. So remember, axons are the long tail-like appendage of your neurons. This is where the action potential gets propagated down, starting at the axon hillock, all the way down to the axon terminal. So white matter typically lies deep to the cortical gray matter. It has this pearly white color due to the high concentration of myelin around the nerve fibers. It's composed of what are known as tracts, which are bundles of axons that connect one part of the brain to another, as well as to the spinal cord. So here we have both gray matter and white matter, and remember that APR will highlight the structure indicated. So the orange here is basically overlaid on the gray matter, and then the orange here is overlaid on the white matter, which you can easily see on this other image. All of this light color, that's all white matter. So it's just being highlighted here so you can see the relationship between gray and white matter in the brain. Okay, the meninges. You might be familiar with this from our very first week of class when we were talking about cavities of the bodies and membranous linings of those cavities. Meninges are the lining of the brain and the spinal cord. So in the brain, there are 3 connective tissue membranes, and that's the same pattern that you see in the spinal cord. They are the dura mater, the arachnoid mater, and the pia mater. Dura, think durable. That's going to be the really tough, fibrous outermost layer that is your barrier between the actual bone of the cranium and the soft matter of the brain. So what do the meninges do? They protect the brain, and they help to provide a structural framework for the arteries and veins that are supplying blood to the brain. So you will notice if you have an intact meningeal layer on your sheet brain that there will basically be an outline where the arteries and veins are within that meninges once you remove it. As I said, the meninges basically function as a barrier between the nervous tissue and the bone of the cranium. Why that's important should be pretty obvious, but it's just to protect the delicate, complex neurons and associated tissue. It's to protect that tissue from being in direct contact with the bone material, the osseous material. So that helps to prevent injury to the brain. So if we look at a little cross-section here, this is a little cross-section showing parts of both cerebral hemispheres. What this is showing is the longitudinal fissure. So this is a structure known as the falx cerebri. That is only in the longitudinal fissure, so it's this material here. Here we have the bone of the skull, of course, so these are the parietal bones. We have the dura mater being the outermost layer here, which is technically comprised of two different layers, the periosteal and the meningeal layer. Then we have the arachnoid mater. Arachnoid, think arachnid, arachnophobia. It's kind of a web-like structure, so kind of reminiscent of a spider web. And then the pia mater, which is almost indistinguishable from the surface of the cerebral cortex itself. So it's essentially a microscopic film on top of that cortex. And you will see how the arachnoid mater penetrates into this longitudinal fissure as well. So it can be quite difficult to remove, very connective, fibrous tissue type. So cranial meninges, again, this is also showing the fact that you have meninges on the spinal cord itself, showing the fact that it borders the longitudinal fissure and surrounds the entire brain within the cranial cavity. Okay, let's talk a little bit about ventricles and cerebrospinal fluid. Ventricles are the internal chambers or spaces within the brain. So there are 4 of these. There are 2 lateral ventricles, one in each cerebral hemisphere, so one in the right part of the brain, one in the left part of the brain. And we have a very small structure, it's a hole. Remember that foramen, that means hole. Formerly, we learned that in the context of bones and bony landmarks. So we have the interventricular foramen. It's a tiny pore that connects to the 3rd ventricle and allows for the flow of cerebrospinal fluid from the lateral ventricles to the 3rd ventricle. The 3rd ventricle is a narrow medial space beneath the corpus callosum, and the cerebral aqueduct runs through the midbrain and connects the 3rd to the 4th ventricle. And then the 4th ventricle is a small triangular chamber between the pons and the cerebellum, and it connects to the central canal that runs through the spinal cord. So we basically have connective ducts, or basically little canals between all of these structures that allows for the continuous flow of cerebrospinal fluid. Something that we'll see in the sheep brain in addition to the ventricles are the choroid plexus. The choroid plexus is just going to be the spongy mass of blood capillaries on the floor of each ventricle. So it's one of the structure types that tends to maintain its color fairly well. It's going to be darker, kind of pinkish red, and I can point that out when we're doing our dissections. It's basically just the dark material within the ventricles that you'll see. And remember, we have ependymal cells that are a type of glial cell. So it's a type of neuroglia that lines the ventricles, covers the choroid plexus, and the primary role of those ependymal cells is going to be to produce that cerebrospinal fluid. All right, so let's take a look at our ventricles here. Here we have a transverse section showing the lateral ventricles. So remember that this is the corpus callosum. Lateral ventricles are going to be these orange areas. Again, corpus callosum, lateral ventricles are going to be right below that in this coronal section. And then another transverse section showing the extent of the lateral ventricles in this region. We have the third ventricle in a coronal section. It basically looks like a little slit. Lateral section, it's not going to be this entire area, but you'll see the space where that ventricle actually is during a dissection. And then the fourth ventricle is pretty small. It's just this little triangular space located inferiorly and posteriorly to your third ventricle. Choroid plexus, basically this is just showing you the location of the choroid plexus. Again, this is just the blood capillaries that line the bottom of your ventricles. All right, so let's discuss briefly the ventricles as well as the cerebrospinal fluid. The cerebrospinal fluid is often abbreviated as just the CSF. It's a clear, colorless liquid that fills the ventricles and canals of the central nervous system, and it also surrounds the exterior of the brain and spinal cord, for example. The brain produces and absorbs about 500 milliliters of this fluid a day, but only 100 to 160 milliliters are normally present at any given time. About 40% of the CSF is formed in the subarachnoid space that's external to the brain. 30% is formed by the general ependymal lining within the ventricles, and then 30% by the choroid plexuses themselves. So production of cerebrospinal fluid begins with filtration of blood plasma through the capillaries of the brain. Ependymal cells are going to slightly modify this filtrate, so the CSF has a little bit more sodium and chloride than blood plasma does, but it has less potassium, calcium, glucose, and very little protein. So it is essentially a watery solution with some dissolved solutes. So this just shows the flow of cerebrospinal fluid. It starts here with number 1. So the cerebrospinal fluid is secreted by the choroid plexus in each of the lateral ventricles. So the lateral ventricles basically right inferior or almost within the corpus callosum. Then the CSF is going to be secreted through, rather it flows through the interventricular foramina, the holes, into the third ventricle. So this is the lateral ventricle. This is the interventricular foramen, which is essentially just a very small hole that allows that CSF through it. And then this space down here with number 3 is the third ventricle. So the choroid plexus within that third ventricle is going to add more CSF. Then you have the flow of your cerebrospinal fluid down the cerebral aqueduct into the fourth ventricle. So this space right here, all the way down here to the fourth ventricle, is the cerebral aqueduct. Your fourth ventricle, again, is just this little kind of triangular area between your brainstem and your cerebellum. Number 5, your choroid plexus within the fourth ventricle adds still more cerebrospinal fluid. And then number 6, we have the CSF flowing out the 2 lateral apertures and 1 median aperture. Finally, we have this space that essentially surrounds all parts of the brain. We have CSF filling the subarachnoid space and bathing the external surfaces of the brain and spinal cord. And then finally up here at 8, at the arachnoid villi, the CSF is reabsorbed into venous blood of dural venous sinuses.